Good day, ma'am. Together here with Kaye Julia now, we will gonna discuss about the different stages of photosynthesis. So here in the illustration, we have two different stages of photosynthesis. We have the light-dependent reaction and the light-independent reaction. So first, in the light-dependent reaction, we have what we call the chlorophyll. These are the green pigments that will usually be found in the chloroplast of the plant. And it functions to absorb light coming from the sun. So we have what we call the H2O, or the water, as seen in illustration. It slowly enters the chloroplast, and it is where the process of photosynthesis takes place or starts. So after that, the process or the reduction reaction process will happen here in the inside of the thylakoids, and we have the end product here, which is what we call the H2O, or the oxygen gas. The first phase of the photosynthetic process is the light-dependent reaction. Here, light energy is trapped and converted into chemical energy by the thylakoids of the chloroplast. This process is aided with the different pigment molecules that forms dusters known as the photosystems, as shown in the illustration. We have two photosystems work together during the light reaction. We have the photosystem 1, or the PS1, or also known as the PS700 because it is most reactive to 700 nanometers wavelength of light. And secondly, we have the photosystem 2, or the PS2, or otherwise called as the PS680 because it reacts most in the 680 nanometers light wavelength. They are termed PS1 and PS2 based on the order of discovery. However, the sequence in which they will proceed with the light reaction is from PS2 to PS1 as shown in the diagram here. Each photosystem contains a cluster of chlorophyll and accessory pigments. Two photosystems vary in electron transfer chain as here, located adjacent to them. The light reaction starts with photosystem 2 absorbs light, which excites the electrons here in the reaction center chlorophyll, then captured by the primary electron acceptor. The reaction center must have a continuous replacement for electrons that jump out and energized by light. An enzyme associated with PS2, the photosystem 2, splits water molecules that allow the electrons to escape and serve as replacement electron in the reaction center. Once the electron is captured by a primary electron acceptor, it is passed through the first molecule of electron transfer chain in photosystem 2, while the electron moves from one another electron carrier molecule to the next. Its energy is being depleted. Some of this energy is released and used to pump up hydrogen ions, as seen here, ions from stroma to the thylakoids compartment, where they will utilize to generate ATP, as shown here, in the process known as the chemiosmosis. The electrons leave the electron transport system with very low energy and enter the action center of photosystem 1, where they gain energy when the pigments absorb solar energy. The excited electrons are passed with primary electron acceptor photosystem 1 and along the electron transfer chain of the PS1 until they reach the nicotinamide nucleotide phosphate, or the NADP+. So an electron carrier together with the electron, NADP+, picks up hydrogen ions from the water to become NADPH. The products of light reactions are the energy carriers ATP and ADPH. Oxygen is released by a byproduct. So ATP and ADPH will be later on used in the Calvin cycle, as it will be discussed by our partner, Kaye Julia Nang.
So I am Kaye Julia Nang, and I will be discussing the second process of the photosynthesis, which is the Calvin cycle. So first, the carbon dioxide enters in the stomata of the plant, which can be found in the leaves of the plant. That's where also the oxygen also goes out from the plant. The CO2 needs to be reduced into sugar such as glucose. So the reductive process needs to happen somewhere in the stroma of the chloroplast of the plant. So the NADPH which is a product from light-dependent reaction, will be oxidized again back into NADP plus, giving its electrons to CO2 or the carbon dioxide. So the CO2 receives those electrons, turning into glucose. During this process, the ATP, which is also a product from light-dependent reaction, needed to power the Calvin cycle. So as the ATP converts back into ADP plus phosphate, it energizes the Calvin cycle, giving it the energy needed to convert the CO2 into glucose. So the reactants here in this stage of photosynthesis are the CO2, ATP, and NADPH. And the products are the sugar such as glucose, and the ADP plus phosphate, and lastly the NADP plus.
Alright, let's continue with the next step of photosynthesis, which is called the light-independent reactions. This is also referred to as the Calvin cycle, if you recall from our previous discussion. The whole purpose of light-independent was to produce these two molecules, which is the NADPH and the NATP. Now, unlike the light-dependent reaction, which takes place inside the thylakoid and along the thylakoid membrane, the Calvin cycle takes place primarily in the stroma of the chloroplast. So that's where we will begin. So, the first process of the Calvin cycle is the carbon fixation. Carbon fixation is the initial incorporation of carbon into a larger organic molecule. This reaction is catalyzed by the enzyme rubisco, which attaches a carbon dioxide molecule with a ribulose biphosphate, or known as the RUBP molecule. This produces 6 carbon molecule that quickly breaks down into 3 phosphoglycerate, 3 GPA molecules, which is the reason why Calvin cycle is often referred as the C3 pathway or the carbon pathway. This process requires 2 molecules of ATP.
And in the second stage of the process of Calvin cycle, ATP and NADPH are used to convert the 3 PGA molecules into molecules of 3 carbon sugar glyceraldehyde 3-phosphate, or the G3P. The 2 molecules of G3P are converted into 2 molecules of phosphoglyceraldehyde, or the G3P, a 3-carbon compound. G3P is the first stable product of photosynthesis. It needs 2 molecules of NADPH. And the third stage of the process is the regeneration of RUBP. By means of enzyme catalyzed reactions, some G3P molecules go to make glucose, while others must be recycled to regenerate the RUBP acceptor. The RUBP is generated from G3P for the process to continue. A molecule of ATP is needed for the regeneration process. The remaining G3P molecule, which is the end product of photosynthesis, exits the cycle. The three turns of Calvin cycle are necessary to produce one net gain of G3P. 5 PGA molecules are required to generate 3 molecules of RUBP. The 3 turns of Calvin cycle use up 9 molecules of ATP and 6 molecules of NADPH. G3P is a substance used by plants to make other organic substances. An example of these organic substances synthesized by plant is glucose. To make one glucose molecule, a 6-carbon sugar, 2 molecules of G3P are linked. This means turns of Calvin cycle. So this is where the Calvin cycle ends, and the end product of the overall process is the glucose.